Good morning, students. Today we will discuss anatomy of number 1, dicot root, number 2, monocot root, dicot stem, and monocot stem. We will discuss the anatomy of all these four with the help of diagram. Let us first study anatomy of dicot root. So if I draw a dicot root, the first layer which we see is of epidermis. So this is the first layer which we see and it is known as epidermis. So this layer is epidermis. Now epidermal cell protrude out in the form of root hairs. So these are the protrusion of epidermal cells known as root hairs. After the epidermis, there are cells which comprise the cortex. So next layer is of cortex. So now with red color, I am drawing the cortical cells. So these are the cortical cells. Let us draw them completely. The cortical cells are thin-walled parenchymatous cells and they have intercellular space in between. So these are the cortical cells which I am drawing in red color. So these are the cells of cortex, and as I've already told you, they are parenchymatous cells. Parenchymatous cells, and they have intercellular space in between. Now the next layer is of endodermis. Endodermis consists of barrel-shaped cells with intercellular space. So now I am drawing the endodermis. So this is the endodermal layer, endodermis, which consists of barrel-shaped cells. So they are barrel-shaped cells, and these cells do not have any intercellular space. Now the wall of endodermis has deposition of suberin. So I am drawing in yellow color the deposition. This yellow color I am drawing is the deposition of suberin. Deposition of suberin in the endodermis. So this is the deposition of suberin in the endodermis, which consists of barrel-shaped cell and without any intercellular space. Due to this deposition of suberin, which is a waxy layer, the endodermis is impermeable to water. Next comes the next layer, which is known as pericycle. It is formed by thin parenchymatous cells. So next layer that I am drawing is a layer of thin parenchymatous cells, which is known as, this layer is of pericycle. So this layer is of pericycle, which consists of parenchymatous cells. Now you should know that initiation of lateral roots and vascular cambium during secondary growth. During secondary growth, when lateral roots and vascular cambium develops, its development take place in the pericycle layer. Again I am telling you that initiation of lateral root and vascular cambium during secondary growth take place in the pericycle layer. Now comes the vascular bundle, which are the xylem and phloem. So here I am drawing the xylem and phloem. This is the xylem which I have drawn. This is the xylem, and now I'm drawing phloem with black color. So these are the phloem. This is the phloem. These are the vascular bundles found in dicot root. The arrangement of xylem and phloem in the dicot root would be radial and exarch. So the arrangement would be radial and exarch. The arrangement of vascular bundles are radial and exarch. Radial means that the vascular bundles are arranged in different radii, in different directions, and exarch means protoxylem towards the periphery and metaxylem toward the center. So protoxylem would be towards the periphery and metaxylem would be toward the center. Now, in between these xylem and phloem, there is a conjunctive tissue which is made up of parenchymatous cell. So there is also a conjunctive tissue between xylem and phloem which is made up of parenchymatous cell, and the central portion is known as pith. You can see that in dicot root, this pith area which I am drawing in black color now is very small and inconspicuous. So this was anatomy of dicot root. Now let us talk about monocot root. Monocot root has same layers like first epidermis, then would come the cortex, then would come the endodermis, then would come the pericycle, then the vascular bundles, which are the xylem and phloem, and then the pith. But there are two important differences. Two important differences between monocot root and dicot root. They are, the first difference is that in monocot root there are more than 6 xylem bundles. More than 6 xylem bundles. This more than 6 xylem bundles is known as polyarch xylem. So the monocot root have polyarch xylem because they have more than 6 xylem bundles. And second difference is that pith in monocot root is large and well developed. You know that pith which we have talked in the dicot root, it was very small and less developed, whereas this pith in the monocot root is very large and well developed. And of course, because monocot root do not have cambium, vascular cambium, they do not undergo secondary growth. Now let us study about dicot stem. Dicot stem. We have covered the roots and now we are talking about the dicot stem. If we look at the diagram of the dicot stem, the first layer again is the epidermis. The first layer would be the epidermis. So this layer is epidermis. Now, similar like in roots, epidermis gave some projections which were known as root hairs. Similarly, in stem, the roots will give some projections, and these projections are known as trichomes. So these are the trichomes, which are the epidermal cell projections in the stem. After the epidermis, the next layer in the dicot stem is the hypodermis, which is made up of collenchymatous cells. So next layer which I am drawing is hypodermis layer, which is made up of collenchymatous cells. So this layer is the hypodermis, which is made up of collenchymatous cells. It is made up of collenchymatous cells. After the hypodermis, below the hypodermis, there is a layer which is known as cortex. And this cortex, which I am drawing in blue color, is made up of parenchymatous cells with intercellular space. So next layer comes the cortex, which is made up of parenchymatous cells with intercellular space. So these are the parenchymatous cells with intercellular space. So this is the layer after the hypodermis, which is cortex, which is formed from parenchymatous cells. After the cortex comes the next layer, which is known as endodermis. Now I am drawing endodermis in yellow color. So next layer is the endodermis. You should remember that the endodermis layer is rich in starch, so it is also known as starch shell. So this is the endodermis, and it is rich in starch, so also known as starch sheath. The next layer is of pericycle. Pericycle is present in patches and made up of sclerenchyma. So now I am drawing the pericycle with green color. So next layer is the pericycle. It is present in patches. So this is the layer which is known as pericycle, and it consists of sclerenchymatous cells. After the pericycle, there would be vascular bundles. So here are the vascular bundles. Now, because it is a stem, this is the phloem, this is the xylem, this would be the phloem, this would be the xylem. Because it is a stem, so vascular bundles would be conjoint, means xylem and phloem in one radii. They would be endarch. Endarch means xylem, metaxylem towards the periphery and protoxylem towards the center. The vascular bundles in dicot stem, again I am writing here for your convenience, they are conjoint, means xylem and phloem in one radii, and they are endarch, means protoxylem toward the center and metaxylem towards the periphery. Now in between the vascular bundles, there are some cells which are known as parenchymatous cells, which are known as medullary rays. So here would be some parenchymatous cells, and these cells would be known as medullary rays. These cells are the medullary rays. Which are present between the two vascular bundles, xylem and phloem. After that comes the last layer, which is known as pith, which is formed from parenchymatous cells, which I am drawing in green color. But these parenchymatous cells are of the pith. The green parenchymatous cells which I am drawing are of the pith. The last layer is the pith. So this is the pith. So this is the anatomy of dicot stem. The first layer is epidermis, which has projection called trichomes. We have called hypodermis, which is collenchymatous. Then we have the cortex, which is parenchymatous. Then we have endodermis, which has starch deposition known as starch shell. Then we have the pericycle, which is made up of sclerenchymatous cell. Then between the two vascular bundles are the medullary rays, and last layer is pith. Vascular bundles are conjoined and endarch. Now let us study monocot stem. If we look at monocot stem, the outer layer again would be of epidermis. So the outermost layer is of epidermis. Inner to epidermis would be hypodermis. But now this hypodermis is not collenchymatous like the dicot stem. It is now sclerenchymatous. So this is the hypodermis, which is made up of sclerenchymatous cells. So we are drawing some layers of the hypodermis. So this is the sclerenchymatous hypodermis. Now comes the cortex. Cortex consists of parenchymatous cells. So this is the parenchymatous cortex. I'm drawing it. Parenchymatous cortex. So these are the cells of parenchymatous cortex. Now in between these cortical cells would be present the scattered vascular bundle. In monocot stem, the vascular bundles are scattered and they are surrounded by sclerenchymatous bundle sheath cells. So now in red, these are the bundle sheath cells which I am drawing. So these are the second bundle sheath cell which I am drawing. These are the bundle sheath cells which surrounds the xylem and phloem. Xylem and phloem. So bundle sheath cells are surrounding the scattered vascular bundle. Bundle sheath cells, and these are the vascular bundles which are surrounded by bundle sheath cells, and these vascular bundles are scattered inside the cortex. Vascular bundles are again conjoined, means xylem and phloem in the same radii, and they are closed. Closed because a cambium would be absent because it is a monocot stem, so it will not undergo secondary growth. This is the diagram of anatomy of monocot stem. It has epidermis. It has a sclerenchymatous hypodermis as compared to collenchymatous hypodermis in dicot stem. It has scattered vascular bundles, and the vascular bundles are surrounded by bundle sheath cells.